Hello students, myself School of Pharmacy, Suresh, Gyan, Vihar University, Jaipur. In physical pharmaceutics second, now today we have to discuss about the micromeritics topic based on methods for determining particle size. In, in previous lecture, we discussed about optical microscopy method and sieving method. And in today's lecture, we have to discuss mainly sedimentation method and particle volume method. So, objective of today's lecture to study about the different particle size determination methods like sedimentation method for particle size determination, its method, its procedure, principle, and lastly, advantages and disadvantages of sieving method. Similarly, in second type of method, which is based on measurement of particle volume, so we have to discuss about the principle, measurement, procedure, apparatus, then lastly, advantages and disadvantages of particle volume measurement method. So these are the two methods which we have to discuss in our today's lecture. A student will be able to know about sedimentation method and particle volume measurement method after completion of today's lecture. Now, as we discussed in previous lecture, method for determining particle size. So everyone know that the particle sample or powder sample having different particles of different sizes. So they are termed as polydispersed powder. And as the particles having different size ranges, so we cannot determine or we cannot say that the powder sample having all the particles of uniform size. So there are so many methods available which can be used for determination of average particle size or particle size of powder samples in terms of average particle size or frequency distribution or weight distribution. So only four methods which is mentioned here we have to discuss, and two methods is already discussed in our previous lecture part 1, and in second part of micromeritics method for determination of particle size we have to discuss about sedimentation and particle volume measurement method. In optical microscopy, we discussed that it can be applicable for the 0.2 to 100 micrometer size of particles, having the so many advantages and disadvantages because it is a direct method. It is also known as number distribution method. So by counting the number of particles, we can determine the particle size of given powder sample. In sieving method for particle size determination, it can be applicable for solid powder samples in the range of 40 to 9500 of pore size of particles. It is very simple method and widely used in laboratory practice for pore size particles. In third method, sedimentation method, the particles having the size range into the 0.02 300 micrometer can be used for determination of particle size by the help of sedimentation method. And last method is particle volume measurement method. This is also known as Coulter counter method, or it can be known as electric method or conductivity method, which can be applicable within the range of 0.5 to 300 micrometer particle size. So these are the four methods for determination of particle size distribution for any sample of powder or as well as dispersion. So these are the different approximate size ranges for different methods for particle size and specific surface analysis. Now, after the detailed discussion of optical microscopy and sieving method in our previous lecture, in today's lecture we have to discuss about the very important method, sedimentation method, and next one is the particle volume measurement method. So first we have to start from sedimentation method. So as per the name given, this is the method which is based on sedimentation phenomena. Sedimentation is nothing but it is the settling of particles under the force of gravity. So by measuring the terminal settling velocity of particles through a liquid medium in a gravitational centrifugal environment using Anderson apparatus, we can determine the particle size. So first of all, we have to know about the what is the velocity of sedimentation, what is the rate of sedimentation, and how it affected by different physico chemical parameters. So the sedimentation method is generally applicable for 0.08 to 300 micrometer particle size. And by measuring the velocity of sedimentation, we can determine the particle size of different dispersion or suspension systems. So the application of ultracentrifugation to the determination of molecular weight of high volume and the particle size in the sub-sieve range can be obtained by gravity sedimentation as expressed in Stokes law. So very important law which is related with the sedimentation phenomena. As everyone knows, this is the Stokes law or theory of sedimentation is based on Stokes law. So under the force of gravity, when we prepare a suspension, the particles can settle down after some time. And the rate of sedimentation or velocity of sedimentation can easily expressed by the help of very important law which is known as Stokes law. It is written like the h by t or v equal to h by t is equal to d square ht rho s minus rho 0 multiplied g divided by 18 eta 0. And when we again rearrange this equation in terms of determination of Stokes diameter, dst is equal to under root 18 eta 0 h divided by rho s minus rho 0 gt, where equation number 1 can be helpful to determine the velocity of sedimentation, and second equation is helpful to determine the diameter of particular particle or particle size can be determined with the help of second equation. So both of the equations are based on the Stokes law of sedimentation. So in this formula, when we go through the detail regarding the Stokes law, we can find out what are the different parameters given in the Stokes law. So where V is the rate of settling. In this equation, V is the velocity of sedimentation, or it is known as rate of settling. And H is the distance of falling time T. So in this formula, you can find the V equal to H by T, where V is the velocity of sedimentation or rate of settling down. And H is the distance of falling time T, whereas dst is the mean diameter of the particles based on the velocity of sedimentation. It is also known as Stokes diameter. Rho s is the density of the dispersed phase particles, and rho 0 is the density of the dispersion medium. So rho s minus rho 0 is the difference in the density of dispersed phase particles and dispersion medium. And g is the acceleration due to gravity. It is 9.8 meter per second square or 980 centimeter per second square. And eta 0 is the viscosity of the medium. So this equation holds exactly only for spherical particles falling freely without hindrance and at a constant rate. So by the help of this equation, we can determine, or we can say that there are so many factors which affects the rate of sedimentation or velocity of sedimentation. Like if the particle size is increased, the velocity or rate of settling down of particles will be also increased because it is directly proportional to the V. And rho s minus rho 0 is the density difference between the dispersed phase material and dispersion medium. So if it is more, then it will be rate of settling will be more. And eta 0 is inversely proportional to the V. So eta 0 is the viscosity of dispersion medium. And if the viscosity of the dispersion medium decreases, the velocity of sedimentation increases. And if the viscosity increases, then velocity will be vice versa decreases. So it is the inversely proportional to the rate of settling. So these are the different parameters which can be obtained or which can be explained by the Stokes law, which affects the rate of sedimentation or velocity of sedimentation. So the law is applicable to irregularly shaped particles of various sizes as long as one realizes that the diameter obtained is a relative particle size equivalent to that of the sphere falling at the same velocity as that of the particles under consideration. So when we go through the equivalent spherical diameter, as we discussed in our previous lectures, if the rate of sedimentation of asymmetric particle is equal to the rate of sedimentation of spherical particle, then the diameter of spherical particle will be the equivalent Stokes diameter for asymmetric particle. So we can obtain the rate of sedimentation or particle size with the help of Stokes law for different types of particles. And very important instrument which can be used in this method, it is known as Anderson pipette method. So you can see the diagram of Anderson pipette apparatus here. Now, when we go through the sedimentation method, the particles must not be aggregated or clumped together in the suspension. So because it is widely used for suspension, and we can determine the particle size of suspension type of dosage form of dispersion systems by the help of sedimentation method. So the particles must not be clumped or aggregated together because such clumps would fall more rapidly than the individual particles and error in results can be obtained. So for accurate results, we have to try to separate out the particles. We can take care that the particles must not be aggregated or clumped together in the suspension. We can use the proper deflocculating agent which must be found for each sample that will keep the particles free and separate as they can fall through the medium. We can obtain the accurate results. So proper deflocculating agent must be used in this experiment. Now sample preparation and analysis conditions. So in this method of particle size determination, the particle size can be determined by examining the powder as it sediments out. In cases where the powder is not uniformly dispersed in a fluid, it can be introduced as a thin layer on the surface of the liquid. So if the powder is not uniformly dispersed in the dispersion medium, it can be seen as a thin layer on the surface of the liquid. If the powder is diophobic in nature, dio means solvent, phobic means heating. So if the powder is diophobic in nature, like the hydrophobic in water, it may be necessary to aid dispersing agent to aid wetting of the powder. And dispersing agents are the surface active agent, or we can use the wetting agent, which are also surface active agents. So by the help of these type of surface active agents, we can easily mix or solubilize the powder in given solvent if it is diophobic in nature. In case where the powder is soluble in water, it will be necessary to use non-aqueous liquids or carry out the analysis in a gas system. So these are the different sample preparation and analysis conditions for sedimentation method for particle size determination. Now, principle of measurement. How can we determine the particle size distribution? How can we measure the particle size of given sample with the help of sedimentation method? So, particle size analysis by sedimentation method can be divided into two main categories according to the method of measurement used. And what are the two type of categories? Let us see here. One of the type of it based on the measurement of particle in a retention zone. Another type uses a non-retention measurement zone. So particle size analysis by sedimentation method can broadly divided into two main categories. One is based on the measurement of particle in a retention zone. Another one is based on the particles in a non-retention measurement zone. An example of non-retention zone measurement is known as the pipette method. In this method, non-volume of suspensions are drawn off and the concentration differences are measured with respect to time. And one of the most popular of the pipette method developed by Anderson and Lundberg and commonly called as the Anderson pipette method. So only we have to discuss about the Anderson pipette method because it is very important and it can easily provide the results for measurement of particle size. So in non-retention zone measurement, we have to use the pipette method generally for determination of particle size. So several methods based on the sedimentation are used. Principal among these are the pipette method, balance method, hydrometer method. So there are different methods based on the sedimentation phenomena we can use for determination of particle size for given dispersion, like the pipette method, balance method, and hydrometer method. So only the pipette method will be discussed here because it combines ease of analysis, accuracy, and economy of the equipment. It is more easier than the other method, and the analysis of sedimentation measurement is more easy compared to other method. It provides the more accurate results, and the economy of the equipment is comparatively less than the other method. So the Anderson apparatus method is widely used for determination of particle size by the help of sedimentation method. It usually consists of a 550 ml glass vessel containing a ml pipette sealed into a ground glass stopper. So a 20 ml pipette is inside the ground glass stopper and the capacity of this vessel is 550 ml. When the pipette is placed in the cylinder, its lowest part or lowest tip is 20 centimeter below the surface of the suspension. So this is the diagram of Anderson pipette apparatus. So Anderson apparatus for determining particle size by the gravity sedimentation method can be seen here. This is the 550 ml glass vessel where a 10 ml pipette is given here, and the lowest part of this tip is around 20 centimeter from the height of the suspension, and a glass stopper is here. So this is the Anderson apparatus for determining particle size by gravity sedimentation method. Now, sedimentation method or procedure. The analysis is carried out in the following manner. Like, first of all, at 1% or 2% weight by volume suspension of the particles in a medium containing a suitable deflocculating agent is introduced into the vessel and brought to the 550 ml mark. So, as we discussed, we have to use a deflocculating agent which can be helpful to segregate the particles or which can prevent the aggregation of different particles. So first of all, we have to prepare 1% or 2% weight by volume suspension of the particle in a medium containing a suitable deflocculating agent, and the sample should be poured into the glass vessel and brought to the 550 ml mark. The stoppered vessel is shaken to distribute the particles uniformly throughout the suspension, and the apparatus with pipette in place is clamped securely in a constant temperature bath. So after pouring the sample, we have to shake the vessel so that there will be uniform distribution of the sample throughout the suspension and the apparatus, and pipette should be placed and it is clamped securely in a constant temperature bath. At various time intervals like maybe 5 minutes or 10 minutes, what is suitable and given. At 10 ml samples are withdrawn and discharged by means of two-way stroke probe. So after a specific time interval, we have to separate out 10 ml sample with the help of the pipette, and then the samples are transferred into a petri dish and it is evaporated, and then after evaporation it is again weighted or analyzed by other appropriate means and correcting for the deflocculating agent that has been added. So first we have to prepare the sample, transfer into the 550 ml glass vessel and again shake it. After shaking, at various time intervals, we have to take 10 ml samples through the pipette and then it should be transferred into a petri dish, and then the samples are evaporated and then we have to wait and analyze the sample, and we have to discard the volume or amount for deflocculating agent that has been added. The particle diameter corresponding to the various time periods is calculated from the Stokes law, as we discussed in equation for Stokes law, and it is determined with the help of H in equation. So it is the height of the liquid above the lower end of the pipette at the time of each sample is removed. The residue or dried sample obtained at a particular time is the weight fraction having particles of sizes less than the size obtained by Stokes law. Calculation for that time period of settling. So by the help of Stokes equation, we can determine the weight fraction of particles. So after transferring the sample into petri dish, and when it is completely evaporated, then we have to weigh the sample and determine the weight of residual material or dried material. And this dried sample obtained at a particular time is the weight fraction having particles of size less than the size obtained by the Stokes law calculation for the time period of settling. And this is known as particle under size. So the weight of each sample residue is therefore called the weight under size, and sum of the successive weight is known as cumulative weight under size. So after drying the sample, when we weigh the sample of residual material is called as weight under size, and all the collection or summation of the successive weights is known as cumulative weight under size. It can be expressed directly in weight units or as percentage of the total weight of the final sediment. Such data can be plotted in different figures like weight distribution or frequency distribution curve. The cumulative percentage by weight under size can then be plotted on a probability scale against the particle diameter on a log scale in the statistical diameters obtained as explained previously. So we can obtain the different graphs from sedimentation method of particle size determination. So this is the very simple important method for determination of particle size determination. It is known as weight distribution method, which is based on the weight distribution of different powder particle sample. Now, after the sedimentation method, which was totally based on Stokes' law, we have to discuss about the particle volume measurement method. So this is the fourth method for particle size distribution. So we can measure the particle size in the range of zero point 0.5 to 300 micrometers. So we can say that the very fine particle size can be determined with the help of particle volume measurement method. This method has different names like the conductivity method or Coulter counter method, because the Coulter counter apparatus is the apparatus which can be used for measurement of particle volume, and we can indirectly calculate the particle size of given powder, given sample of dispersion. Now, in this type of machine, the powder is suspended in an electrolyte solution. The suspension is then made to flow through a short insulated capillary section between two electrodes, and the resistance of the system is measured. So this is the principle of this particle volume measurement method. First, we have to prepare the dispersion or suspension, and this suspension is then made to flow through a short insulated capillary section between two electrodes. And what is the resistance generated after the passing the suspension, we have to measure. When the particle passes through the capillary, there is a momentary peak in the resistance, and the amplitude of the peak is proportional to the particle size. So when the resistance generated due to the particles, the amplitude of the peak is proportional to the particle size, and counting is done by automatically with the help of a computer. So this is the most advanced sophisticated method which can be used in pharmaceutical industry as well as in our laboratory practice for determination of particle size distribution. So this is in this PPT, the range of analysis with the help of particle volume method is given here. We can determine the particle size of very simple solution. Now, how can we prepare the sample and how can we analyze the particle size with the help of particle volume measurement method? So, sample preparation and analytical conditions which is required for particle volume measurement method. So, powder samples are dispersed in an electrolyte to form a very dilute suspension. The suspension or dispersion is usually subjected to ultrasonic agitation for a period to break up any particle agglomerates. A dispersant may also be added to a particle de-agglomeration. So this is the conditions which is required for sample preparation and analysis through the particle volume measurement method. First, we have to prepare the dispersion in an electrolyte solution to form a very dilute dispersion. And then the suspension or dispersion is usually subjected to ultrasonic agitation for a period so that it can be easily helpful to break out the particles and segregate the particles. We can aid the dispersant to prevent the agglomeration. So these are the different conditions required in this particle volume measurement method. Now, principle of measurement is we discussed. The particle suspension is drawn through an aperture accurately drilled through a sapphire crystal set into the wall of a hollow glass tube. So it is the apparatus where the electrodes are put in the sample dispersion and the resistance developed when the particle pass through this, we have to measure it. So in detail description of the principle of the measurement of particle volume measurement method. The particle suspension is drawn through an aperture which is accurately drilled through a sapphire crystal set into the wall of a hollow glass tube. Electrodes situated on either side of the aperture and surrounded by an electrolyte solution. They monitor the change in electrical signal which occurs when a particle momentarily occupies the orifice and displaces its own volume of electrolyte. The volume of suspension drawn through the orifice is determined by the suction potential created by a mercury rate rebalancing in a convoluted tube. The volume of electrolyte fluid which is displaced in the orifice by the presence of a particle causes a change in electrical resistance between the electrode which is proportional to the volume of the particle. So this is the basic principle of measurement. The volume of electrolyte fluid which is displaced in the orifice by the presence of a particular particle, they causes a change in electrical resistance between the electrode and which is proportional to the volume of particle. And by knowing the volume of the particle, we can convert this volume into the particle size by different equations. So the change in resistance is converted into a voltage pulse which is amplified and processed electrically. Then pulses falling within pre-calibrated limits or thresholds are used to split the particle size distribution into many different size ranges. In order to carry out size analysis over a wide diameter range, it will be necessary to change orifice diameter used to prevent the coarse particles blocking a small diameter orifice. So when there is a chance to damage the orifice, then we have to change the orifice diameter to prevent the coarse particles blocking a small diameter of orifice. Conversely, finer particles in a large diameter orifice will cause too small a relative in volume to be accurately quantified. Now, how can we measure the particle size? Let us see here. The popular instrument for measurement of the particle volume is known as the Coulter counter apparatus. So what is Coulter counter? This instrument operates on the principle that when a particle suspended in a conducting liquid passes through a small orifice on either side of which are electrodes, a change in electric resistance occurs. So the principle of Coulter counter is the similar as the particle volume measurement determination method. So this instrument operates the same principle that when a particle suspended in a conducting liquid passes through a small orifice on either side of which are electrodes, a change in electric resistance occurs. In practice, a known volume of a dilute suspension is pumped through the orifice, provided the suspension is sufficiently dilute. The particles pass through essentially one at a time. So it should be very carefully considered that each particle should pass through separate time through the orifice, and we can select the suspension so that the particle pass through the essentially one at a time. This is the diagram of a Coulter counter used to determine the particle volume. So in this diagram, you can see here there is the electrolyte solution and particles is given here. Then counter switch is given here. Then mercury is used to measure the different concentrations, and by the help of the mercury manometer, we can determine the difference after the passing the particles through this electrode solution. Then there is an amplifier to amplify the signals, and pulse height analyzer is given here. So pulse height proportional to the particle volume, and a digital register is given here which can maintain the different number of particles, and it is related with the counter drive. So this is the schematic diagram of a Coulter counter apparatus which can be used for determination of particle volume. And when the electrolyte solution pass through the electrodes through the orifice of this apparatus, there is a generation of electric resistance, and this electric resistance can be analyzed with the help of pulse height analyzer, and it is recorded and maintained in digital register. So a constant voltage is applied across the electrodes to produce a current, and as the particles travel through the orifice, it displaces its own volume of electrolyte, and this results in an increased resistance between the two electrodes. The change in resistance, which is related to the particle volume, causes a voltage pulse, and that is amplified and fed to a pulse height analyzer calibrated in terms of particle size. So as we see in our diagram, the pulse height analyzer, which can be useful, and it can be provides the different signals which is already amplified with the help of amplifier. The instrument records electronically all those particles producing pulses that are within two threshold values of the analyzer. By systematically varying the threshold settings and counting the number of particles in a constant sample size, it is possible to obtain a particle size distribution. So, with the different varying the threshold settings and counting the number of particles in a constant sample size, we can obtain the readings in terms of particle size distribution. This instrument is capable of counting particles at the rate of approximately 4,000 per second. So you can see here the capacity of this instrument is very large. We can easily calculate so many particles, and the speed is very high in case of particle volume measurement method by Coulter counter apparatus. So both dose counts and particle size distributions are obtained in a relatively short period of time. So this is the very big advantage of this Coulter counter apparatus. The data may be readily converted from a volume distribution to a weight distribution. First, we have to obtain the result in terms of volume distribution, and then it will be converted in terms of weight distribution. So this is the again simplified diagram where you can see the particle pass through the orifice, and there is a generation of electrical resistance. And this electrical resistance is directly proportional to the particle size. So by measuring this electrical resistance with the help of Coulter counter apparatus, we can easily determine the particle size. So advantages and disadvantages of this method. So we already discussed that this method is more accurate or precise than the other method. Analysis range is wide comparatively from other methods, we can count more than 4,000 particles in a second, as we discussed in Holter counter apparatus. So these are the different advantages which makes the particle volume measurement method or Holter counter method more efficient than the other type of methods. Then what is the disadvantage or what are the limitations of this Holter counter apparatus method? So it is a sophisticated method as we see in description, and it is also an expensive method. We require a learned helper or labor cost of this method is higher than the other methods. So beside the disadvantages, this method is more precise and more widely used for analysis of particle size in a dispersion system. So these are all about the today's methods. So in summary, we discussed about different methods for particle size distribution for any sample, whether it may be present in powder form or maybe in the dispersion form. So in today's lecture, we discuss mainly sedimentation method, which is based on sedimentation phenomena or Stokes law, and the second method, which is based on particle volume measurement. And the apparatus for this method is known as Holter counter apparatus. And how it is applicable and how it is used, we discussed in our today's lecture. So these are the some of the references for this particle size determination method by the help of sedimentation or particle volume measurement. So you can go through these books and find out more details about these two methods. Now, quiz section. So after completion of today's lecture, the student may be able to give the answers of the following questions. The question number 1 is given here. Any instrument used for measuring volume of particle is. So we have to give the correct answer from ABCD option, where the options are given like Anderson apparatus, Holter counter, Fisher, subsieve sizer, and zeta meter. So which instrument is used for measuring volume of particle? We have to write the correct answer. Then sedimentation rate can be decreased by. So how the sedimentation rate can be decreased? So after completion of today's lecture, the student may be able to give the answer of this question. There are 4 options. One is given here: particle size increase, then particle size decrease, unchanged particle size, or not possible to change. So we have to go through the sedimentation method and find out the correct answer of this question. The next question is given here. Anderson apparatus works under the principle of. So options are microscopy, sedimentation, sieving, or Holter counter. So Anderson apparatus method or Anderson apparatus works under the principle of which method? We have to give the correct answer from ABCD option. The next one is the Fisher subsieve sizer, is mainly used for particle surface area determination, particle surface volume determination, particle size determination, or solubility rate determination. We have to pick the correct answer from A, B, C, D options. The next question is example of number distribution method is. So we already discussed what is the weight distribution method, what is the number distribution method. So you have to go through the lesson or find out the correct option for number distribution method, whether it may be microscopy, sedimentation, sieving, or rheology. Next question is weight distribution method used for particle size determination is. So again, we have to give the example of weight distribution method for particle size determination. A option is given here, sedimentation. B is sieving. C is the optical microscopy, and D is the sieving and sedimentation both. So we have to find out the correct option for weight distribution methods used for particle size determination. And when we go through the different short answer type questions, These are the different questions like the discuss the factors affecting sedimentation. So what are the different factors which affects the rate of sedimentation or velocity of sedimentation? Next one is the what is Stokes law. So what is Stokes law and how it is expressed? You have to give the answer after studying the today's lecture. Then enumerate the methods for particle size determination. So as we discussed, there are four methods for particle size determination. You have to go through the lecture and find out the correct options. And last question is the give the advantages of alter counter method. So we discussed alter counter method in very detail. So you have to write the advantages of alter counter method for this question. So this is all about the today's lecture. Please go through the lecture and find out the all the methods for determination of particle size distribution of different sample. So okay, thank you.